In this video, we will teach you how to use a pH meter.
To calibrate the pH meter, we opened the bottle of the pH meter and rinsed it in distilled water. After shaking off the water from the meter, we placed the probe in pH 4 buffer and calibrated the meter to read a pH of 4. Then we rinsed the meter off with distilled water. We repeated this process with the pH 7 buffer and again calibrated the meter. After rinsing the pH meter off with water, we tested the pH of both buffer solutions to ensure that the meter was correctly reading the pH.
After calibrating the pH meter, rinse the pH meter with distilled water. Place the pH meter in unknown solution and record the pH reading. Place the meter in distilled water after cleaning it. At the end of the lab, rinse the pH meter with distilled water, dry the meter, and place it back in the bottle which it came from.
A buffer is a solution that resists changes in pH when small amounts of acid or base are added to it. Buffers are typically made out of a weak acid and its conjugate base, which is the acid's anion. When an acid is added to the buffer, it is absorbed by the conjugate base. When a base is added to the buffer, the dissociated hydrogen ions react with the hydroxide ions to create H2O.
A strong acid will completely dissociate in water, so it will produce a high concentration of hydronium. A weak acid will only partially dissociate in solution, so it will produce relatively low concentration of hydronium. Because the dissociation of hydronium is so small, the dissociation reaction is easily reversible. This means that the weak acid dissociation will have an equilibrium reaction. To minimize the effects of equilibrium, it is assumed that the initial concentration of hydronium is 0 molar, and the change in concentration for the acid is small enough to be negligible, so the initial concentration remains constant.
Be sure to record your data.